Well, thank you for coming. I'm Lew Lim, as you know, and I will be talking about COVID, acute, and then going to long COVID. Right. Disclosure: I am a shareholder of Vielight, so there's a conflict of interest. But I try to be as objective as possible. Sometimes I cannot avoid because Vielight is like the only device out there. Now, go back to when COVID first started. I don't know whether it's kind of embedded in the memory. Do you remember how it all kind of unraveled around the beginning of 2020? And you were hearing stuff going on overseas from China, and US was trying to figure out what to do. And then the first lockdown happened, I think in January or early February 2020 in Kirkland, Washington State, right? There's a retirement home. Everybody panicked, everybody got infected. So that was kind of like the story of February 2020. But I'm showing this for a reason. In the same month, there was this cruise liner, Diamond Princess. If you follow the news, people were watching, and there's this ship that was docked in Yokohama, and they did not let anybody out of the ship for like 2 weeks. And people not, and it was, it was a new thing, so people didn't know what to do. I show this because we had one passenger who owned a Vielight intranasal, and we were communicating by email. And he was saying, people are just throwing off like flies around us. It's a panic. I don't know what to do, but I got a Vielight. I feel protected. I feel healthy. Now, I know 712 passengers, crew members infected. There were 14 deaths. I can't say whether it was the device that kept him healthy, but this thing really got us going. We got going because there's panic around. Looks like someone was benefiting, and someone on another ship, I think it was docked in LA, also reported something similar. So say, okay, we really ought to do something. We have no time to develop a new device, but we had one ready at a time. It was called the X Plus. Why the X Plus? Okay, you can position it on the chest where the thymus gland is to reduce T cells, to help to heal, and then the intranasal. So we got it going. We did a white paper. I got the support of Michael Hamlin from Harvard at the time. So yeah, let's do this, and so we did. To cut the story short, we designed a study. There's a whole war stories about recruitment and all that stuff. We eventually were able to analyze two groups. One, there were 99, eventually 99 subjects who were on the usual care, which is like, okay, you got this, stay at home, stuff like that. Get yourself, do this, the test. Then the 100 were on active. So these were the results for the acute study. And we just about squeaked through with a significance of 0.05. And the primary endpoint was, and there was no other, we did our own research, and there was no other test for recovery, but our endpoint was how fast they were recovering from the infection. So we had this, it's called the WURSS 44 from University of Wisconsin. I think it's Wisconsin Upper Respiratory Symptoms Survey. Now what we found, this is in summary, the primary endpoint. So we showed significance from the hazard ratio. You may not be able to see, this is the probability of not recovering, and those on active were significantly lower, trending lower than the control. But it was actually applicable to 0 to 7 days of infection at the severe stage. But they recover, and then we published a paper. We submitted for approval. Now, these are the secondary endpoints. There were a bunch of them. These are the significant ones. They're involved in headache and a bunch, other things. I just mentioned that I highlighted brain fog, which is think clearly, because it's going to lead us to our next thing. And we got the approval from Health Canada to treat that, and they extended the approval to treat upper respiratory tract infection, which cover things like COPD, chronic obstructive pulmonary disease, and cold, flu, bunch of things, because it basically treats the upper respiratory tract infection. We argued that point. Now, that study finished in 2022. It didn't end because people were being affected in a long spell, right? The long COVID. And you look at, they look healthy. Some of them are on wheelchair. And it was something that, acute COVID was what we treated before. And I say, okay, it's not going to be a thing anymore. Actually, today, nobody cares about acute COVID, right? It's still infection out there. People are landing in the hospital, but it's kind of like flu. So it's not a thing anymore. So, but at that time, okay, we got it we have some kind of a solution for acute COVID, but long COVID was there, and it was a new thing. Now, and I thought, and some of you might know somebody who is long COVID. Is pretty nasty. So these numbers come out of the US, actually the National Center for Health Statistics. The last set of data they had, these are the latest numbers, August 2024. And I think that's all you're going to get, because I think the administration they're not funding them anymore. So that's all you're going to get. What they showed was actually 17.9% of people who had been infected with COVID before ended up with long COVID. And it's estimated to be 17.6 million in the US. Globally, I think it's something like 400 million. And the numbers actually pretty much stay the same. The numbers have not come down. Now, what's going on? What's causing long COVID? This was only just published last year. It took them a long time. It's a high-level committee, the National Academies of Science, Engineering, and Medicine Committee. So they want to try to put their brains together. What is it? Actually, these facts are already been around. It's been discussed. It's pretty obvious, but they needed a committee to put it official. There are a bunch of symptoms. Long COVID has like 200 symptoms, but I say the two main ones are fatigue and brain fog. Now, what are the implications? People are not able to work, so many of them are on disability. They have problems caring for themselves, so many of them are at home. There are emotional problems like depression, and the families are affected. Then I want to go to brain fog, but the numbers are not clear. You get numbers. This is a UK-based group, so it's like 25 to 75%. It's so wide, but you just know that a lot of people are affected with brain fog. And the main there is no treatment for brain fog, actually, medication, even for long COVID, as you probably know. But there is more clarity, more momentum in coming up with cognitive rehabilitation programs. It's like graduated mental exercises. Actually, the evidence is not clear. They haven't nailed down what the program is going to be. It's still in discussion. So it's not actually resolved by any means. Now, has neurofeedback done anything? So I did a search. I don't practice neurofeedback, so I want to find who has done any work and published anything on long COVID. Now, there's one study. This one is actually, these two studies are based on this one single subject, a 34-year-old woman. And I think they were looking at F34, C34, P34, and saw significance in F3 and P3 and C4. But one case, then they went deeper with the same woman. So there's just one case. I don't agree necessarily with their, if the method they used, okay, you do the training, but the method they used to evaluate whether this person is recovering from brain fog. So I think they use like self-reporting, whether actually one woman, and also the clinical IQ, I think. There was another one based on Osmer method, infraslow, and intra-hemisphere method, but it actually was just, outcomes was in anxiety and depression. So brain fog is just one. What do I think? I think there's potentially just one person. Sometimes it's really hard to do any study on more people, So I just give some benefit of doubt here. Now, brain fog in long COVID was a hard to figure out issue. There were so many studies coming out from many people, but I think it settles down to these few things. It's mainly inflammatory based. You get this infection and your immune system respond. You get into a situation, it may get into, it affects the oligodendrocytes, which is your that supports your axons, the myelin sheath, hippocampal degeneration, so you get brain fog and so on. There are discussion that is autoimmune. I'll talk about it more later. And then there is this infection in the brain. They did find traces of virus in the brain through imaging, through actually autopsy actually. And then Epstein-Barr virus could be activated. Then we got the blood-brain barrier being compromised, and hypoxia and others, affecting the circulation. There was a couple of people looking at imaging, and they did this tracer and found that, it's not just confining to the olfactory bulb, medulla, brainstem, and cerebellum. Actually, it's pretty widespread. Now, there is similarities in the symptoms between long COVID and Alzheimer's disease. I just want to bring it up, mainly because we have done quite a lot of work on Alzheimer's, and we have devices that are, designed to treat Alzheimer's disease. So there will be, just to keep it short, the infection actually causes the release of inflammatory factors, getting into this cycle of reactive oxygen species and affecting the mitochondria, go into a loop, and then it affects the the heart, the lung, and actually the brain. So it can lead to cognitive dysfunction. So to summarize, we are looking at the similarities in neuroinflammation, the cytokines, activated microglia. Here are the biomarkers. The phosphorylated tau found as biomarkers in Alzheimer's disease in the neocortex and the cerebellum, right? Then there is this movement in mainly in Toronto, in Canada. Donald Weaver is a professor at the University of Toronto. He is really working on. He came up with this theory that Alzheimer's disease is, and I mentioned that long COVID has something to do with the immune system. It's autoimmune. It keeps, is your body attacking itself with the antibodies and so on. It keeps doing it. So he reckons that Alzheimer's disease is an autoimmune problem. He said it's a natural process that your brain produces beta amyloid to fight this abnormal expression in your brain, but it is only when it becomes misfolded or the tau get also a protein also get misfolded, hyperphosphorylated as they call it, that it is a problem, and that is it becomes that immune system has got into the stage of autoimmune, so you get this problem. Okay, fine, that is his theory. Though that allows us to draw similarities, the overlap between Alzheimer's disease and brain fog. So shared biomarkers like elevated pro-inflammatory cytokines, oxidative stress, mitochondrial dysfunction, these biomarkers, beta amyloid and the tau. Actually, this is very key. Today's presentation, I'm not talking about the fundamental mechanisms because just don't have that much time. But a fundamental mechanism is on mitochondria. And actually, in my opinion, this expression of the biomarkers are because of the dysregulation in mitochondria. And if your intervention can resolve at the mitochondrial level, it can help with all this stuff down the line. So if I have time, I'll talk about Alzheimer's disease, one of my favorite topics. Now, so this brain fog, and I actually listened to one of her presentations, and she was actually suffering with long COVID. She's a professor at McMaster University, and she said, I used to read like 16 papers a day, and now I could hardly go through one. But her lab did this study on 106 people and found these markers of inflammation in the body. So the IL-1B and the other ones are actually markers of inflammation. So she said, okay, this is where things are pointing to. Now, then it leads to, do we have an intervention? I think photobiomodulation could be an intervention because this was actually released during COVID. This was done in Iran, and he had there was some, I think there were 10 patients. They were testing it with lasers, right? It looks like this is the back of the patient, and that's where his shirt was put up, and they were directing lasers, low-level lasers. And these are the inflammatory markers that are quite typical. You're talking about IL-6, this is TNF alpha. This is a good guy, IL-10, and then you have the other one, TNF alpha. or IL-8, okay. IL-6, 4, and TNF alpha. Actually, IL-10 is a good marker. So they did, then you say, okay, how much of, because the red is with photobiomodulation and the gray is placebo. Those were infected, is up, and then and they all, even the, what do you call it, they all get reduced, including the IL-10, which is the good guy. So they did this ratio and compare IL-6 and IL-10. Looks like the ratio is in favor of, having more IL-10. Then there is discussion, could be a lymphatic flow issue. Your lymphatic system in your body, in your brain is not clearing. Now, there was a proposal to do this like kind of massage method to help the lymphatic flow, and they argue that it really helps. Okay, so lymphatic system is something to think about. And then photobiomodulation has been for quite a while, talking about, but these are discussion, this is called inferred, coming up with a hypothesis that photobiomodulation helps with the lymphatic flow, so it clears the stuff that you don't want in your brain or your cerebral spinal fluid. Now, these are all discussion, and what do I have here? Okay, so 2 days ago, this group that we are working with, Baycrest Hospital, attached to the University of Toronto. Now, this is actually part of a bigger study that we're involved in. They're doing real-time fMRI, real-time EEG, 128 channels, healthy subjects, but very few people have managed to see what's happening to the brain treated with photobiomodulation in real time. Like, there's always pre and post, clinical results and so on. So this is a really important study. A lot of things are going to come out. So we did also computer simulation, which supported it. But this is one of the first things that come out, the lymphatic flow. We did they did part of a big study looking at the different skin color. There is the detector for skin color. There's one laser to the head, one of the nose, and this was the MRI scanner. Now, we explored different frequencies, 808, 1064, those who are familiar. Some in LEDs, there are 810 and 1070, the popular ones. And different power, and different pulses, 10 hertz and 40 hertz. And they did 4 minutes just collecting data, 4 minutes treatment with photobiomodulation, these guys here, and then 4 minutes later and see what's happening. Now, the focus is actually on the fourth ventricle. Why the fourth ventricle? The lymphatic system into the brain are in different ventricles. The fourth one is the one that connects your brain to the spinal cord. So that was it's like a gateway. So that's why they focused on. That's what it looks like in imaging. And they were looking right through the fourth ventricle here. I will start with the bottom line. Photobiomodulation done this way actually increased the lymphatic flow. So until this time, we all thought hypothesizing, bringing on different other studies and say, yeah, it might do that. Now, something unique is happening here. It's never been discovered. What it does is the inflow was slow. Then you get this swelling, which they detected through the imaging. And then when it goes out, it's fast. The net is actually fast. And I was debating about the hypothesis with them. It looks like photobiomodulations is almost like developing a pumping action. It's just like heart, build up the blood from coming back from the vein and forcing it out in the arteries, like a water pump. You build it up and then you press it out. So they're exploring it, but this is very interesting. Just to summarize, it's significant. And the wavelength that has more effect is 808 for laser, which is 810, more than 1064. On LED is often cited at 1070. And the peak was like 100 milliwatts. So you need quite a lot of power density to have this effect. This bigger paper is going to confirm it and all that. So too low density doesn't do that much, but that's not the point of this particular study. The intranasal, when I was talking 100 milliwatts here, both 808 and 1064, the effect was achieved by intranasal with only 5 milliwatts. So that is the significant thing. It will be confirmed with some other papers they're coming up with. So bear in mind, these little portable things that we have on intranasal, actually they're pretty powerful. Then gamma, as in Alzheimer's disease, here is with actually with looking at EEG, eyes open, eyes closed, eyes open and eyes closed, EOEC somewhere. But looking at the healthy young population, healthy age, older population, the elderly population, and Alzheimer's disease. Actually, okay, as you age, your gamma, when it was like peak at 37.5 for young adults, it was actually lower at 35 or something, and the peak are lower. But in Alzheimer's disease, gamma is almost not there, there's a peak at 30, but the amplitude is lower, the power is lower. So it's good to have gamma for Alzheimer's disease, and we can draw across to long COVID. So in summary, we want to calm an overactive immune system. We want to control inflammation. We want to address this Alzheimer type of biomarkers and improve lymphatic flow. Try to induce endogenous gamma because that is correlated with healthy versus Alzheimer. And we have already kind of drawn parallels between Alzheimer's disease and long COVID, right? Then we have this. And basically the parameters are just that gamma, which is 40 Hertz, deliver that. The default mode network, I didn't mention it, but default mode network has been implicated in Alzheimer's disease. So we're trying to rebuild a default mode network, the intranasal, mention it, lymphatic junction. So when we position it here, there are actually major junctions at here above the nasal area. So we're pointing at those places. And we want to do a study, a double-blind study. And for practical reasons, you want to translate to, widespread use to resolve this problem, home use. And so we do remote data collection. This is the usual thing you submit for a console diagram, but I just want to say that we assessed 90 subjects for eligibility. Quite a challenge. But with all this dropout and not meeting protocols, we ended up with 21 subjects and 20. They're allocated to actually down to the sham and active group. So we have a device that's sham. They take home, they don't know whether it's real or not. And then they're active. Now, what did we find? We did 56. The primary endpoint was looking at these time intervals, 14 days, 28 days, 56 days. And then we did a follow-up post-treatment. Now, how do we treat for brain fog? We actually went through quite a number of different techniques to try to tease it out, actually for long COVID, many researchers are using the NIH toolbox. We looked at the toolbox, we thought this was better. Krios, it is Krios Research. The company was called Cambridge Science. Right, got it. And these tasks that we are testing them on were mainly focusing on collectively here as a primary outcome, short-term memory, executive function and reasoning, attention, verbal memory. I made this bold because this one was particularly impactful, feature match. Then we have secondary outcomes looking at grammatical reasoning. Spatial span is a task that you would try to place objects in the right places. Digispan, I think you look at numbers, progressive and going backwards, and see whether you get it right. And odd one out, double trouble, I think involves like Stroop tests. You know, you look at a green and it's labeled as red, and you have to quickly say what it is. So that's one of the, it is to do with this. Some of these are to do with short-term memory and some are attention. So let's see what the results are. Now, these are collective. Actually, I'm still in the process of writing this paper. I got pulled away by this conference. So collectively, we use least square mean. And now this is actually important to note. Unfortunately, it didn't work for everybody. We stratified those who are below 45 years old and those at 45 and above. So collectively, this task didn't really do that much for the elderly population, but very significant for below 45 years old. We can talk about why, the plasticity, and so on. Now we were observing them at various timelines. So the green is the active below 45, and you know these are the below 45 sham, which is orange, and then you have the above 45 active in sham. So when we stop treatment and look at what followed, those who are below 45 continued to improve. You know, there was much elsewhere. There's not much change. Those above 45 didn't. I'll show more of that. Now these are the various tasks in Krios. The blue are those that were 45 and below. Right across the tasks, it was all positive. But the above 45 was very interesting. They were doing really well for this feature match, which is to do with attention, but did pretty badly with tasks that involve short-term memory. But collectively, it came out like like a wash, But although the younger population had lower significance, say, for attention, but collectively, even like for short-term memory, performed very well. So collectively, it did well. Now, this is a table of what was significant. I said the composite cognitive score I showed you, and this particular one that a good p-value, and that this one, the one that was involved in attention, was also significant. So this is like breaking down a little bit. And we also did secondary outcomes that were not dealing with cognition. Right across everybody, the mobility improved, and the actually this is to do with memory too. It's a different test, but the quality of life, let's say, generally improved. So summary, PBM is promising. Why don't I say, oh, it works for sure? But you might argue the population is small, so we might or might not want to do a bigger population. It's really expensive to do a clinical trial, but we're applying to Health Canada to get approval. I don't think FDA will entertain that small number. And the efficacy to reduce brain fog is significant here for 45 and below. That's the conclusion. But here is the thing. There was no reduction in chronic fatigue. So I got all these things with brain fog. Chronic fatigue, I showed you, is one of the two main symptoms. So what do you do? Chronic fatigue is ME-CSF. Myalgic encephalomyelitis is a mouthful, but CFS is easy to remember. Actually, chronic fatigue has been around for ages. It doesn't have to do, to be related to COVID. There's no answer. It's idiopathic. Some of the smartest brains, including the professors, their own children have suffering and bedridden. So there's no clear answer. And so where are the clues? Okay, this is pointing towards maybe a clinical study, There is this vagus nerve infection hypothesis. There's localized infection, that's infecting the vagus nerve and leads to this other cytokine bombardment. So you're talking about inflammation, dysregulation, autonomic dysregulation, reduced heart rate variability, the parasympathetic tone that correlate with fatigue severity. Then it's neuroinflammation. So there's persistent inflammatory factors that disrupt maybe the blood-brain barrier and mitochondrial function. So there are clues here. There is some other studies that continue to point towards inflammation affecting your autonomic system. So the attention is still drawn to fatigue and exercise intolerance. Right now, this is a study that is really interesting. I would show it, and I don't know. Well, it's got accepted in a decent journal. So what they did was they did the nasal stimulation, some mechanical stimulation. You just wave this stimulator, and your nasal area senses it. But they've arguing that it is a form of dealing with the chronic fatigue. And well, I'll just focus on, because some of the things don't add up, but I'll focus on the neurostimulation that does it. So the nose area is actually receptive for something, and this was a temporary effect for them actually to help with chronic fatigue. There are actually quite a number of studies that proposes vagus nerve stimulation. Many of them non-invasive is the auricular one. This one is to do with the auricular stimulation. And it actually brings down the inflammatory factors. And so there's other discussion about vagus nerve to treat with epilepsy. We did discuss very briefly with other people. Bunch of other things like anxiety, headache, and so on. But here we're talking about possibly helping with chronic fatigue. So there are pop articles, Scientific American. Usually they're written by quite good journalists. They're proposing that look, chronic fatigue could respond, and I'm looking for those that link long COVID chronic fatigue. So there are a couple of articles. This is also proposing, it's a small study, that vagus stimulation can possibly treat long COVID chronic fatigue. But everybody says, oh, we need a big study, we need something big. So, and I did, you point to this QR code, you get to my white paper that I did to position photobiomodulation as a method for vagus nerve stimulation. I have this table, but you get it in the paper. But I compared with what it can do, and I compared with auricular. They're both is electrical stimulation, or the other big. These are non-invasive electrical stimulation on the vagus nerve on the neck. If you haven't got this, come to me later. I'll get you the link. So in summary of when I propose it, it is photobiomodulation doesn't give you any sensation of tingling. If you use electrical stimulation, the higher power you can feel more and more, and then you got to put a gel. And I've got anecdotal study. I've got anecdotal. I wouldn't say studies, experimentation internally. I've got a study coming out of Harvard, and they have tested it for themselves. Of induced stress. We've got possibly starting one with another university for PTSD, and they all kind of experimented on themselves. So, okay, let's try in big clinical studies. Then they all measure with heart rate variability. You can do HRV with stimulation with slow rhythmic breathing, 6-7 times a minute. It could also work. You can try this as well with vagus nerve stimulation. You could do electrical, or you could do this particular one. But we have studies coming out for stress, PTSD, seizure risk, and sports performance. This. Okay, this is what I showed that we are doing at a lot of work. So we got 15 minutes. What we're doing with COVID is just one part. We've got a lot of stuff going on. You can see on the website. I'm saying this because this was done in October. It keeps building up. But we love research. We love to try something, see if we fail, see if we succeed. You never know until you do this stuff. So these are indexed. These numbers are indexed to the work we're dealing with. And I think there are more than 100 researchers who are involved with the various studies we're doing. And so there are a lot of people, and not just one guy. We've got a team of talented people. And I tell you, some of these researchers we're working with are brilliant. They are actually well-renowned worldwide as well. So I love working with them, but watch out for what's coming. Thank you.